Assalamu alaikum dear students. Welcome to the course Principle in Animal Life 1. This is lecture number 42. The topic is Substrate Specificity of Enzymes. Discussing agenda for the topic will be enzyme substrate complex, active site, and induced fit. Lecture outcome. After watching and listening this lecture, students will know about the interaction between an enzyme and its substrate. Enzymes are biological catalysts. They speed up the chemical reactions by lowering the activation energy, whereas substrate is anything upon which the enzyme acts. You can say substrates are the reactants which convert into products, and this particular reaction is catalyzed by the enzymes. The reactant an enzyme acts on is referred to as the enzyme's substrate. So substrate is anything upon which the enzyme acts, and in a chemical reaction, reactants and products are involved. So the reactants are referred to as the enzyme's substrates. The enzyme binds to its substrate or substrates when there are two or more reactants, forming an enzyme substrate complex. So what is an enzyme substrate complex? When the enzyme binds with the substrate, so their combination forms enzyme substrate complex. While enzyme and substrate are joined, the catalytic action of the enzyme converts the substrate to the product or products of the reaction. So once the enzyme substrate complex is established, then these enzymes helps the conversion of those substrates or those reactants into products. The overall process can be summarized as follows. Here you can see enzyme binds with the substrate and enzyme substrate complex is established, and then the products are formed, and enzymes are not used in a chemical reaction. This is one of the biggest property of the enzymes, that they convert reactants into products, but they are not themselves used in this reaction. They are available to catalyze other reactions. Most enzyme names end in ase. So ase is a common ending point of the enzyme name, just like ligase, DNases, topoisomerases, helicases, etc., lactase. So these A's are usually the last three alphabets of an enzyme's name. But it is not a hard and fast rule, because there are some exceptional cases where the last three alphabets of the enzyme does not end with the word ase, just like the ptyline is one enzyme. For example, the enzyme sucrase catalyzes the hydrolysis of the disaccharide sucrose into its monosaccharides glucose and fructose. So here they have given an example of this condition where the last three alphabets of an enzyme's name ends with A's. But remember, this is not a hard and fast rule. So sucrase is an enzyme which is responsible for the conversion of sucrose into glucose and fructose. First, the sucrase binds with sucrose and enzyme substrate complex is established, and then the products are formed. The reaction catalyzed by each enzyme is very specific. An enzyme can recognize its substrate even among closely related compounds. Now, this specificity is very important because the living organism is like a container in which thousands of chemical reactions are going on. So, for the purpose of regulation, the enzymes are highly specific. They only catalyze a specific reaction and they doesn't interfere with other reactions going on inside the cell. So this specificity is very important for maintaining a proper order inside the cell or the organism. For instance, sucrase will act only on sucrose and it will not bind to other disaccharides such as maltose. So this is example of the specificity of the enzymes, that they only catalyze a specific chemical reaction because the enzymes has an active site where the substrate binds. And this three-dimensional structure of the enzyme, and in particular the structure of its active site, is responsible for determining the specificity of an enzyme, that this particular enzyme will only catalyze those reactions. This particular enzyme has that substance as a substrate. What accounts for this molecular recognition? So what is responsible for this specificity? Why sucrase cannot catalyze the reaction of maltose? Why only sucrose? Recall that most enzymes are proteins. Yes, all enzymes are not proteins because recent discovery suggests that RNA can also act as an enzyme, and such enzymes are called ribozymes. So now we cannot say that all enzymes are proteins, but majority of them are proteins. Recall that most enzymes are proteins and that proteins are macromolecules with unique three-dimensional structure. When we were discussing proteins, and right from the beginning of this course, we are saying again and again that one of the important rules in biology is that structure determines the function, and that statement holds true for proteins more than any other molecule. And here is yet another example. The three-dimensional structure of an enzyme and its active site determines or decides which reactions it is going to catalyze. The specificity of an enzyme results from its shape, which is a consequence of its amino acid sequence. So because the structure of an enzyme is determined by the sequence of nucleotides in DNA. The sequence of nucleotides in DNA corresponds to the sequence of amino acids in a polypeptide, and then in a polypeptide chain, when there is twisting and folding occurs, then the amino acids tends to come face to face with one another, and then interactions are established. So all is determined by DNA, and then in turn the sequence of amino acid, that which type of a folding will occur in that protein and that polypeptide. Only a restricted region of the enzyme molecule actually binds with the substrate. So only a specific area, a particular area of an enzyme is in contact with the substrate, and that area is called the active site, which is typically a pocket or groove on the surface of the enzyme where catalysis occurs. So this is the three-dimensional structure of an enzyme, and it is the space-filling model where all the atoms forming this enzyme have been represented. So this is the active site where the catalysis occurs, and this is the substrate which will interact with this active site, and then the reaction will go forward and products will be formed. So this particular enzyme's substrate is glucose. So this is one of the enzymes that is used in glycolysis. It is a hexokinase. Usually the active site is formed by only a few of the enzyme's amino acids, with the rest of the protein molecule providing a framework that determines the shape of the active site. So here only few amino acids are present around this area. The bulk of the amino acids form the outer boundaries of this enzyme, and it is these enzymes in that particular areas which determines this particular shape. The specificity of an enzyme is attributed to a complementary fit between the shape of its active site and the shape of the substrate. So in order to catalyze a reaction, in order to convert a substrate or a reactant into product, the first thing and the most important thing is the establishment of enzyme substrate complex. When this complex is not established, or unless this complex is established, there will be no reaction. So the establishment of this complex is of predominant significance, and this is something which determines the specificity of an enzyme, because everything cannot fit into that active site. There will be a certain specific substance which will fit here, and then this enzyme will catalyze the reaction. So this pocket or this groove and something which fits here perfectly determines the specificity that this particular enzyme is only there to catalyze that particular reaction. An enzyme is not a stiff structure locked into a given shape. Some of the enzymes, their structure is flexible, not a rigid structure. So here we are introducing another concept which is induced fit model. In fact, recent work by biochemists has shown that enzymes and other proteins seem to dance between subtly different shapes in a dynamic equilibrium, with slight differences in free energy for each pose. So what they want to say is that the recent research suggests that the structure of an enzyme is not fixed, it's not rigid, but it can oscillate between two or three different shapes, like a pendulum oscillates between a central point. Therefore, each enzyme can oscillate into two or three different shapes, and a dynamic equilibrium is established between those oscillations, and each shape has some free energy. Free energy is that energy which can perform work, which is available to perform work. So these different structures in which the enzyme oscillates, they all those structures have slight difference in the free energy, or in the amount of energy possessed to perform some work. Obviously, enzymes perform work by catalyzing reactions. The shape that best fits the substrate isn't necessarily the one with the lowest energy. But during the very short time the enzyme takes on this shape, its active site can bind to the substrate. So just like a keychain, when you want to introduce a new key into a keychain, then you open that keychain, you apply some force, you open it, and then you introduce the key, and when you leave the spring or the keychain comes to its original position. So the stable position of that keychain was a closed one, and when you introduced your force, then this shape of the keychain changed, and then you introduced the key, and then when you released it, it returned back to its original position. So the stable position of the keychain is the closed position. Similar is the case with the enzymes. The enzyme can oscillate between two or three different shapes. It can adopt two or three different shapes. Some of the shapes are energetically feasible, energetically stable, others are not. The active site itself is also not a rigid receptacle for the substrate. So this active site is also not rigid. Actually, when we talk about the enzyme substrate complex, then two, or you can say theories or hypotheses that comes to the mind. The first is the lock and key model, which says that the active site of the enzyme is rigid, and enzyme can only catalyze those reactions the substrate of whom can fit into the active site. The other model is the induced fit model. Induced fit model says that a slight change or modification can occur in the active site of the enzyme in order to accommodate the substrate perfectly. So as shown in figure 1b, when the substrate enters the active site, the enzyme changes shape slightly due to interaction between the substrate's chemical groups. Chemical groups on the side chain of the amino acids that form the active site. So why an enzyme's active site changes shape? Because when the substrate comes and it binds to atoms in the active site, it binds to amino acids in the active site, then there is interaction between the R groups of the amino acids and some of the atoms of the substrate. This shape changes makes the active site fit even more snugly around the structure. So this interaction between the R groups of the amino acids in the active site and the atoms of the substrate even more strengthens this association between the substrate and the active site. The tightening of the binding after initial contact, called induced fit, is like a clasping handshake. For example, it's a very good example, like you shake hands with someone and then you hold their hands tightly in your palms. So they say that it is actually the similar kind of situation where the enzyme interacts with active site of the enzyme interacts with the substrate, and then the active site or the protein is wrapped tightly around the substrate. Its active site is wrapped tightly around the substrate. So this is the induced fit. Means a slight change occurred in the active site after the amino acids present in this area. There are groups of those amino acids interacted with the atoms or molecules in the substrate. You can see here when the substrate attached here, that interaction becomes strengthened, and you can say that this enzyme kind of grabbed this substrate like you grab something into the jaws. Induced fit brings chemical groups of the active site into position that enhances their ability to catalyze the chemical reaction. So this particular conformation, it brings the chemical groups of the active site into position that increases the ability of the enzyme to catalyze this reaction. That was the end of the lecture. Thank you very much.